Hi guys, Mr. Pollock Biology here. Again, this is my fourth video in a series looking at this super top secret teacher only AS Biology new specification 2016 specimen material set 2 paper 1, which is the factual recall paper for the new spec. This is the teacher copy. Now, this is the one that's secure from students anyway, that you can't just get off the AQA website. Although someone is distributing copies in the comments of one of these other videos, so you guys can go and ask him. So thank you, mysterious stranger. We're going to look at question 4. We've done question 1, we've done question 2, we've done question 3, and now we're on to question 4, which is a really nice co-transporty, facilitated diffusion-y, biochemical testy type question, which is great. And it's quite a good one. The first part of it is asking about co-transport in the small intestine. So realistically, you guys probably want to check out this video that I've got the link to just above here. That shows things a little bit more visually. Because this is more exam technique, we're just going to be doing words with a couple of sketches here and there. So first of all, we need to say what on earth the ileum is. So that is in the small intestine, just to jog our memory. So cells lining the small intestine of mammals absorb the monosaccharide glucose by co-transport with sodium ions. Explain how. So it's already told us the what, we just want to know the how. So I'm going to draw a quick sketch. The diagram that the animation rather, that I've done on the actual video of this is a lot better, but for the time being, we're going to have our ileum just here, and then we're going to have our bloodstream just here, and in between that we've got a bunch of cells, and those cells we want to get our glucose through and out the other side into the bloodstream, and to do that we use co-transport. But the first thing we're going to look at is why we want to use that system to get as much glucose as possible in. And to keep that system working, we need to make sure we've always got a favourable concentration gradient of sodium. So we want a favourable Na plus gradient from here to here. And then that allows us to utilise this co-transport protein. So the first thing we do is we make sure we've got like no Na plus in here. And we do that by making sure that sodium is not in these cells. We get rid of it into the bloodstream, and then it gets whisked away. So we do that by active transport. So our first mark here is for saying that sodium, or sodium ions, are actively transported out of the cells into the blood. Out of cells into blood. What's the point of that? Well, that maintains a favourable sodium concentration gradient. So maintains an Na plus gradient, and that gradient, of course, we're talking about between from the lumen of the small intestine to the cells, which is great. And then finally, that what we want to say for our third and final mark is what that allows to happen, essentially. So glucose is able to enter, glucose moves with Na plus from lumen to cell, by co-transport, which is a specific type of facilitated diffusion, requires a carrier protein. Facilitated diffusion. And that's co-trans. And that's our 3 marks. Nice and straightforward, really. So watch that video. Hopefully it'll help you understand things a bit more fully. It is in significantly more detail. But this is how we get those 3 marks. So this goes on to talk about an experiment that a student has set up, where we've got some visking tubing that's laid in a, looks like a water bath or a beaker of water, and in the visking tubing we've got starch and amylase. Now amylase is an enzyme that is going to digest starch. Digest that, and as a result of that we will get some maltose. Back to this shortly. So the material from the visking that the visking tubing is made from, we say it's partially permeable, so some stuff can get through and some stuff can't. In fact, it's the big stuff that can't get through. Small stuff passes through no problem. And after 15 minutes, our student has removed samples from the liquid in the beaker and also from the liquid inside the visking tubing. And she's carried out a bunch of biochemical tests on those samples. And we've got to decide what's going on. So let's start with the biorec test. First of all, it's worthwhile saying that the biorec test is the test for protein or polypeptides or the presence of a peptide bond. If you've got any doubts about your biochemical tests, check out my video. It's linked just above here, and it's Know Your Biochemical Tests, and it tells you everything that you need to know. So, protein. Well, do we have any protein? Well, we certainly haven't got any in the beaker. We won't have any even after the experiment's gone on, because the only protein we have is amylase, which is an enzyme. And what are enzymes made from? That's right, protein. So the only place we're going to have a positive result is going to be inside the visking tubing, and it will be a lilac colour because that's the colour that biorec reagent turns. Next up, we've got iodine, which is the test for starch, which is a nice big molecule. And because it's a big molecule, the only place we've got starch is in the visking tubing. It's too big to leave. So again, we're going to have none in the beaker, and we're going to have some in the visking tubing. Now, depending on when we tested this, we might have only a little bit of starch left, the undigested starch, or we might have none. So it's quite a tricky exam question, this one, because you get the mark both if you put a tick or if you don't put a tick, which is kind of bizarre. Final one, Benedict solution. Oh, I should say as well, iodine goes blue-black in the presence of starch, just a bit of revision there. Final one is Benedict solution, which is the test for sugars, or reducing sugars, of which maltose is one. So inside the visking tubing, we will end up with some maltose, but maltose is also able to move out because it's a nice small little disaccharide. So we're going to have ticks in both boxes, and we'll probably have a nice brick red precipitate as a result of that. So all we have to do is put that into words, which is our justification to our answers. So for the biorec test, first thing we're going to say is that amylase is a protein, an enzyme. And then we're going to say that it's too big, however, too large to leave tubing. That's why it's only inside. Then the second mark is a little bit repetitive, so starch again, so the iodine, we're testing for starch. Starch, and again, this one, we can say just the same, it's too large to leave the tubing, so it's going to only remain inside. Where things switch up is the Benedict's test. Because maltose is the product of digestion, this one, let's go for maltose is what we're testing for here. Maltose produced by digestion, let's go for hydrolysis of starch. Now this one is the opposite way round. So this one is small enough to leave the tubing. Small enough. And we'll just poach that to leave tubing. Okay, so that's it. That's our 3 marks again. Again, really nice question, really, I think. So double check my video on biochemical tests. I hope this has been useful for you guys. Please watch the other videos in this series as well. And of course, like, comment, and subscribe. Take care.